5.3 protein interactions modulated by chemical energy, actin, myosin, and molecular motors. Organisms move. Cells move. Organelles and macromolecules within cells move. Most of these movements arise from the activity of a fascinating class of protein-based molecular motors. Fueled by chemical energy, usually derived from ATP, large aggregates of motor proteins undergo cyclic conformational changes that accumulate into a unified, directional force, the tiny force that pulls apart chromosomes in a dividing cell, and the immense force that levers a pouncing, quarter-ton jungle cat into the air. The interactions among motor proteins, as you might predict, feature complementary arrangements of ionic, hydrogen bonding, and hydrophobic groups at protein binding sites. In motor proteins, however, the resulting interactions achieve exceptionally high levels of spatial and temporal organization. Motor proteins underlie the migration of organelles along microtubules, the motion of eukaryotic and bacterial flagella, the movement of some proteins along DNA, and the contraction of muscles. Proteins called kinesins and dyneins move along microtubules in cells, pulling along organelles or reorganizing chromosomes during cell division. An interaction of dynein with microtubules brings about the motion of eukaryotic flagella and cilia. Flagellar motion in bacteria involves a complex rotational motor at the base of the flagellum, see Fig. 19-41. Helicases, polymerases, and other proteins move along DNA as they carry out their functions in DNA metabolism, Chapter 25. Here, we focus on the well-studied example of the contractile proteins of vertebrate skeletal muscle as a paradigm for how proteins translate chemical energy into motion. The major proteins of muscle are myosin and actin. The contractile force of muscle is generated by the interaction of two proteins, myosin and actin. These proteins are arranged in filaments that undergo transient interactions and slide past each other to bring about contraction. Together, actin and myosin make up more than 80% of the protein mass of muscle. Myosin, Mr. 520,000, has 6 subunits, 2 heavy chains, each of Mr. 220,000, and 4 light chains, each of Mr. 20,000. The heavy chains account for much of the overall structure. At their carboxyl termini, they are arranged as extended alpha helices, wrapped around each other in a fibrous, left-handed coiled coil similar to that of alpha-keratin, Fig. 5-27a. At its amino terminus, each heavy chain has a large globular domain containing a site where ATP is hydrolyzed. The light chains are associated with the globular domains. When myosin is treated briefly with the protease trypsin, much of the fibrous tail is cleaved off, dividing the protein into components called light and heavy meromyosin, Fig. 5-27b. The globular domain called myosin subfragment 1, or S1, or simply the myosin head group is liberated from heavy meromyosin by cleavage with papain, leaving myosin subfragment 2, or S2. The S1 fragment is the motor domain that makes muscle contraction possible. S1 fragments can be crystallized, and their overall structure, as determined by Ivan Raymond and Hazel Holden, is shown in Figure 5-27c. In muscle cells, molecules of myosin aggregate to form structures called thick filaments, Fig. 5-28a. These rod-like structures are the core of the contractile unit. Within a thick filament, several hundred myosin molecules are arranged with their fibrous tails associated to form a long bipolar structure. The globular domains project from either end of this structure in regular stacked arrays. The second major muscle protein, actin, is abundant in almost all eukaryotic cells. In muscle, molecules of monomeric actin, called G-actin, globular actin, Mr. 42,000, associate to form a long polymer called F-actin, filamentous actin. The thin filament consists of F-actin, Fig. 5-28b, along with the proteins troponin and tropomyosin, discussed below. The filamentous parts of thin filaments assemble as successive monomeric actin molecules add to one end. On addition, each monomer binds ATP, then hydrolyzes it to ADP, so every actin molecule in the filament is complex to ADP. This ATP hydrolysis by actin functions only in the assembly of the filaments. It does not contribute directly to the energy expended in muscle contraction. Each actin monomer in the thin filament can bind tightly and specifically to one myosin head group, Fig. 5-28c. Additional proteins organize the thin and thick filaments into ordered structures. Skeletal muscle consists of parallel bundles of muscle fibers, each fiber a single, very large, multinucleated cell, 20 to 100 μm in diameter, formed from many cells fused together. A single fiber often spans the length of the muscle. Each fiber contains about 1,000 myofibrils, 2 μm in diameter, each consisting of a vast number of regularly arrayed thick and thin filaments complex to other proteins, Fig. 5-29. A system of flat membranous vesicles called the sarcoplasmic reticulum surrounds each myofibril. Examined under the electron microscope, muscle fibers reveal alternating regions of high and low electron density, called the A bands and I bands, Fig. 5-29b-c. The A and I bands arise from the arrangement of thick and thin filaments which are aligned and partially overlapping. The I band is the region of the bundle that in cross-section would contain only thin filaments. The darker A band stretches the length of the thick filament and includes the region where parallel thick and thin filaments overlap. Bisecting the I band is a thin structure called the Z disc, perpendicular to the thin filaments and serving as an anchor to which the thin filaments are attached. The A band, too, is bisected by a thin line, the M line or M disc, a region of high electron density in the middle of the thick filaments. The entire contractile unit, consisting of bundles of thick filaments interleaved at either end with bundles of thin filaments, is called the sarcomere. The arrangement of interleaved bundles allows the thick and thin filaments to slide past each other, by a mechanism discussed below, causing a progressive shortening of each sarcomere, Fig. 5-30. Figure 5 to the filament. Figure 5 to the thin actin filaments are attached at one end to the Z disc in a regular pattern. The assembly includes the minor muscle proteins alpha-actinin, desmin, and vimentin. Thin filaments also contain a large protein called nebulin, 7,000 amino acid residues, thought to be structured as an alpha helix that is long enough to span the length of the filament. The M line similarly organizes the thick filaments. It contains the proteins paramyosin, C protein, and M protein. Another class of proteins called titans, the largest single polypeptide chains discovered thus far. The titan of human cardiac muscle has 26,926 amino acid residues, link the thick filaments to the Z disc, providing additional organization to the overall structure. Among their structural functions, the proteins nebulin and titin are believed to act as molecular rulers, regulating the length of the thin and thick filaments, respectively. Titin extends from the Z disc to the M line, regulating the length of the sarcomere itself and preventing overextension of the muscle. The characteristic sarcomere length varies from one muscle tissue to the next in a vertebrate, largely due to the different titin variants in the tissues. Myosin thick filaments slide along actin thin filaments. The interaction between actin and myosin, like that between all proteins and ligands, involves weak bonds. When ATP is not bound to myosin, a face on the myosin head group binds tightly to actin, Fig. 5-31. When ATP binds to myosin and is hydrolyzed to ADP in phosphate, a coordinated and cyclic series of conformational changes occurs in which myosin releases the F-actin subunit and binds another subunit farther along the thin filament. The cycle has four major steps, Fig. 5-31. In step 1, ATP binds to myosin, and a cleft in the myosin molecule opens, disrupting the actin-myosin interaction so that the bound actin is released. ATP is then hydrolyzed in step 2, causing a conformational change in the protein to a high-energy state that moves the myosin head and changes its orientation in relation to the actin thin filament. Myosin then binds weakly to an F-actin subunit closer to the Z disc than the one just released. As the phosphate product of ATP hydrolysis is released from myosin in step 3, another conformational change occurs in which the myosin cleft closes, strengthening the myosin-actin binding. This is followed quickly by step 4, a power stroke during which the conformation of the myosin head returns to the original resting state, its orientation relative to the bound actin changing so as to pull the tail of the myosin toward the Z disc. ADP is then released to complete the cycle. Each cycle generates about 3 to 4 piconewtons of force and moves the thick filament 5 to 10 nanometers relative to the thin filament. Because there are many myosin heads in a thick filament, at any given moment some, probably 1% to 3%, are bound to thin filaments. This prevents thick filaments from slipping backward when an individual myosin head releases the actin subunit to which it was bound. The thick filament thus actively slides forward past the adjacent thin filaments. This process, coordinated among the many sarcomeres in a muscle fiber, brings about muscle contraction. Figure 5 to 30. The interaction between actin and myosin must be regulated so that contraction occurs only in response to appropriate signals from the nervous system. The regulation is mediated by a complex of two proteins, tropomyosin and troponin, Fig. 5 to 32. Tropomyosin binds to the thin filament, blocking the attachment sites for the myosin head groups. Troponin is a Ca2+ binding protein. A nerve impulse causes release of Ca2+ ions from the sarcoplasmic reticulum. The released Ca2+ binds to troponin, another protein-ligand interaction, and causes a conformational change in the tropomyosin-troponin complexes, exposing the myosin binding sites on the thin filaments. Contraction follows. Working skeletal muscle requires two types of molecular functions that are common in proteins binding and catalysis. The actin-myosin interaction, a protein-ligand interaction like that of immunoglobulins with antigens, is reversible and leaves the participants unchanged. When ATP binds myosin, however, it is hydrolyzed to ADP and pi. Myosin is not only an actin-binding protein, it is also an ATPase enzyme. The function of enzymes in catalyzing chemical transformations is the topic of the next chapter. Summary 5.3 Protein interactions modulated by chemical energy, actin, myosin, and molecular motors. Protein-ligand interactions achieve a special degree of spatial and temporal organization in motor proteins. Muscle contraction results from choreographed interactions between myosin and actin, coupled to the hydrolysis of ATP by myosin. Myosin consists of two heavy and four light chains, forming a fibrous coiled coil, tail, domain and a globular, head, domain. Myosin molecules are organized into thick filaments, which slide past thin filaments composed largely of actin. ATP hydrolysis in myosin is coupled to a series of conformational changes in the myosin head, leading to dissociation of myosin from one F-actin subunit and its eventual reassociation with another, farther along the thin filament. The myosin thus slides along the actin filaments. Muscle contraction is stimulated by the release of Ca2+ from the sarcoplasmic reticulum. The Ca2+ binds to the protein troponin, leading to a conformational change in a troponin-tropomyosin complex that triggers the cycle of actin-myosin interactions. P